चलिए जल्दी से रिवाइज कीजिए। द नेक्स्ट फिगर 6.1, ए पॉलीन्यूक्लियोटाइड चेन के नीचे जितने भी पोर्शन दिए गए हैं। चलिए द फर्स्ट क्वेश्चन, इन RNA एवरी डैश डैश रेसिड्यू हैज एन एडिशनल OH ग्रुप present at dash dash position in the ribose. Also in RNA, the dash dash is found at the place of thymine. That is dash dash another chemical name for thymine. The next, DNA as an dash dash substance present in nucleus was first identified by Frederick Mesher in dash dash. He named it dash dash. However, due to technical limitation in isolating such a long polymer intact, the elucidation of a structure of DNA remained elusive for a very long period of time. The next, it was only in dash dash that James Watson and Francis Crick, based on dash dash diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous dash dash model for the structure of DNA. The next, one of the dash dash of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition proportion was also based on the observation of dash dash that for a double stranded DNA, the ratio between dash dash and dash dash and dash dash and dash dash are constant and equals 1. इसमें एक वर्ड गलत बोला गया है, बताइए वो वर्ड कौन सा है। चलिए द नेक्स्ट, द बेस पेयरिंग कन्फर्स ए वेरी यूनिक प्रॉपर्टी टू डैश डैश चेन्स। The next, they are said to be complementary to each other, and therefore if the dash dash of bases in one strand is known, then the sequence in other strand can be predicted. Also, if each strand from a dash dash, let us call it as a parental DNA, act as a template for synthesis of a new strand. The two double-stranded DNA, let us call them as daughter DNA, thus produced would be dash dash to the parental DNA molecule. Because of this one dash dash implication of the structure of DNA became very clear. The silent feature of dash dash structure of DNA are as follows. चलिए फर्स्ट नंबर बोलिए, इट इज मेड ऑफ टू डैश डैश where the backbone is constituted by sugar phosphate and the bases project inside. Second, the two chains have anti-parallel polarity. It means if the chain has the dash dash, the other has dash dash. Third, the bases in two strands are paired through hydrogen bond, that is H bonds, forming base pair, that is BP. Adenine forms two dash dash with thymine from opposite strand and vice versa. Similarly, dash dash is bonded with dash dash with hydrogen bond. As a result, as a result, always a purine comes opposite to a dash dash. This generates approximately uniform distance between the two strands of the helix, figure 6.2। माइंड कैप्चर कीजिए फिगर 6.2 का। और इसे नोटबुक पर हमें दिखाइए। जी हाँ, और इसे नोटबुक पर हमें दिखाकर बनाइए। जी हाँ, आमने सामने बनाइए। आई एम प्रेजेंट विद यू। चलिए नेक्स्ट 4। The two chains are coiled in a dash dash fashion. The pitch of the helix is dash dash. A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 meter, and there are roughly 10 base pair in each turn. Consequently, the distance between a BP in a helix is approximately 0.34 nanometer. The next, the plane of one base pair stalks over the other in a dash dash. This, in addition to hydrogen bonds, confers dash dash of the helical structure, figure 6.3। माइंड कैप्चर कीजिए फिगर 6.3 और नोटबुक पर हमें बनाकर दिखाइए। Compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in DNA remain almost constant? The next, the proportion, the proposition of a double helix structure for DNA and its simplicity in explaining the dash dash become revolutionary. Very soon, Francis Crick proposed the dash dash in molecular biology, which states that the genetic information flows from dash dash to dash dash to dash dash। जी हाँ, DNA to RNA to protein। DNA replication dash dash to mRNA dash dash to protein। The next, in some viruses, the flow of information is in reverse direction, that is, from RNA to DNA. Can you suggest a simple name to the process? Thank you so much. You are most welcome in the next session of this discussion series. Please like, share and subscribe.